All right, so chapter 23 is our last chapter where we're going to cover some specific details of a certain biological molecule or group, or class of molecules, and that we'll talk about nucleotide metabolism. So again, another chapter like we had with our protein metabolism chapter where there's not a lot of details we're going to cover for a lot of things. We are going to take a couple of things and look at them in depth. For example, we're going to look at the disease of gout and talk about how that is problematic. We're going to talk about how we use some modified nucleotides as chemotherapy agents or chemotherapeutic agents. And we'll look at the other parts of the process, mainly like in the same way we talked about with protein metabolism, in terms of a substrate cycle, in terms of kind of making or degrading nucleotides, but then also talking about how we metabolize or degrade the component pieces. We'll start the activity kind of the same way that we talked about the last one, except we're just going to add in this next and last piece of these four kind of biological biopolymers that we've talked about was nucleotides. And again, nucleotides are a required building block, but you can synthesize them easily yourself. So we'll talk about kind of some general aspects of that, but we don't really consider them a fuel source. So we've kind of talked about our main three fuel sources in carbohydrates, lipids, and proteins. So we'll spend a lot of time kind of picking apart in our activity this figure. And again, the concept here is thinking about taking ribose 5-phosphate and hooking it with nucleotides. So again, we're going to talk about how we build those nucleotides, and so we'll talk about what's the recipe for all of these, and again, we'll see some important amino acids that we've seen before, for example, in protein metabolism. Do want you to remember what purines and pyrimidines are. And when we talk about the synthesis of these molecules, we'll make sure to kind of discuss that these have different kind of strategies for how they're built. In one aspect, we're going to build the base first, and then we'll attach the sugar to it. And then another one will actually build the base on the sugar. So we'll go into kind of some overview details of that. Kind of some important highlights that we'll talk about here is we'll revisit kind of where we get some of these pieces, for example, the pentose phosphate pathway. We'll talk about the fact that you synthesize ribonucleotide phosphates first, the RNA kind of versions, and then we have important enzymes called ribonucleotide reductases that convert those into the deoxy or the DNA version of that. We see some kind of important species that we've talked about before, malonyl-CoA, so again an important precursor for fatty acid synthesis. And then this is an important guy down here talking about uric acid and the degradative process of purine metabolism. The inability to excrete uric acid is the primary compounding factor in gout. So if you have the inability to excrete this uric acid, it accumulates, and then that can lead to the symptoms of gout. So we'll use that kind of as a medical examination in this chapter. Again, we're going to kind of use this overview of a slide to kind of talk about some details about how we synthesize both purines and then pyrimidines. So I'm not going to go into the details of here. Most of these details we actually aren't going to cover, but we'll again talk about whether or not you build the base first and then add the sugar, or if you take the sugar first and then you build your base on the sugar. So two kind of different strategies for how you make nucleotides. Again, we'll talk about the building blocks that we have for making purines and then for pyrimidines. So again, just flipping through those slides fairly quickly because we'll get to those in our activity. So as I mentioned before, we build the ribonucleotide version first. So again, this is a ribonucleotide where we have a 2' hydroxyl. We use special enzymes called ribonucleotide reductases to take that and convert that into the deoxy flavor. So it's kind of an interesting thing to think about, that the RNA kind of building block is made first, and then we make the DNA versions. And probably a reason that that can be confusing to think about is if we think about the central dogma, we take DNA and we transcribe it into RNA and translate that into protein. So we kind of think about that order of things, and so sometimes that's a little bit confusing to think about. We'll talk about special kind of considerations for deoxy TTP or dTTP. And again, we'll talk about some important intermediates here. We'll see another important aspect of folate. We talked about folate and kind of methylation chemistry as being really important in our protein chapter. So we'll see, again, another B9 importance here. And so we'll talk about that with the synthesis of TTP. And again, we'll spend a good bit of time talking about that metabolic process, the synthesis of TTP, dTTP, and then we'll talk about a few chemotherapy drugs that interfere with this. And the big picture here is to remember that if you interfere with DNA synthesis, you interfere with the ability of cells to replicate, and that's the problem with cancerous cells, is they kind of are replicating out of control. So if you kind of target cells and inhibit their ability to replicate rapidly, you can have an effective chemotherapy agent. So we'll talk a little bit about that, and we'll talk about how some of these different drugs work, whether they're suicide substrates, whether they're what we call competitive inhibitors, and we've talked about those in our kinetics chapter. And then we'll spend just a tiny little bit of time talking about what happens if nucleic acids are in excess. Thinking about digestion of nucleic acids from your food, right? Everything that you eat was probably had a live cell in some way, shape, or form, and so there's DNA that's there. So we'll talk about very minimally how nucleic acids from exogenous sources get into your body. And then very, very generally with the overview of how these species are degraded, kind of the main building blocks we get, and we'll spend a lot of time with this uric acid kind of component, because an inability to excrete uric acid, so we can see here whether we're breaking down A's or G's, and again, hypoxanthine is a derivative of the breakdown product of adenine, we get to uric acid. And if you have the inability to excrete uric acid, then that's problematic because it accumulates in uric acid crystals, and that's very, very painful. So we'll talk about some of the physiological symptoms that you have with gout. But a lot of medicines work to inhibit the production of uric acid. So if you don't produce uric acid, it doesn't matter if you're not able to excrete it. And what actually happens is you open up other channels for metabolizing this xanthine intermediate into more hydrophilic substances that are more easily excreted. Okay, so we'll talk a little bit about that. So again, not a lot of details that we'll learn in chapter 28, or I'm sorry, chapter 22 here, 23, sorry, on nucleic acid metabolism. We're going to focus mainly on some key aspects of what happens when things go wrong with nucleotide metabolism, or thinking about nucleotides is important for cell division, so again, chemotherapy agents.